In this portion of the lecture, we're going to look at protein digestion, absorption, and transport, and then thinking about briefly about sort of evaluating protein quality in the diet. Now, I know prior to this class, in your prerequisite coursework, you've really covered digestion, absorption, and transport of protein. So I'm not going to go into a lot of detail on it, but I would like to just kind of have that fresh in your mind in terms of how those proteins get into the body and absorbed, because when we think from a nutritional perspective, it's great to think about what happens once it's in the body and broken down and how it's utilized, but we also have to remember that everything needs to be functional to actually get that protein in a form that our body can use it. And digestion, absorption, and transport are key in that process. So that's what we'll look at a little bit in this lecture. This image just is, again, a refresher on the protein digestion. And remember that protein really doesn't start digesting until it gets to the stomach. Nothing really happens in the mouth in terms of protein. Once it gets to the stomach, that very acidic environment, the hydrochloric acid, and the gastric juices start the digestive process of proteins. Really, that first step in protein digestion is the denaturation of the protein. So remember, that's removing the quaternary and tertiary structure so that we get it down to really more that primary structure. And then once it starts to move into the small intestine is going to break it down further with hormones and enzymes involved there. So we'll look at a little bit more some of those specific enzymes that function in the small intestine to break those proteins down into polypeptides and amino acids. So this table shows where some of the enzymes are that are involved in protein digestion. On the left lists the zymogens, and remember the zymogen is essentially an inactive enzyme. So when it's in the body, we don't always want it in active form if we don't have a use for it to be breaking things down. So pepsinogen is the first one listed, and that hydrochloric acid or pepsin is what activates it to turn it into the actual enzyme of pepsin. And that's active in the stomach. Again, remember the hydrochloric acid is found in the stomach. We have a lot of hydrochloric acid in the stomach, so that's what activates that and causes that initial digestion of protein, which is, again, really the denaturation. And from that, we're going to get those peptides. And remember, peptides are just amino acids still hooked together, but down more into that primary structure. So we've unfolded them, we've uncoiled them, and now they're kind of just lined up together. So then you see the rest of the enzymes that are listed are really working in the intestines. And the intestines is where the primary digestion of proteins happens to break it down into those smaller parts. So we have trypsinogen, chymotrypsinogen, procarboxypeptidases that all are activated into the active forms of the enzymes and then break down the short peptide chains into amino acids or into sometimes smaller peptide chains. And you can see that there's different substrates for different enzymes. So the enzymes are active on specific amino acids. So for example, the trypsin is active on the basic amino acids. The chymotrypsin is active more on aromatic amino acids and a few others. So which enzyme is going to be acted on or which enzyme, the enzyme is specific to certain types of amino acids, I guess is the way I wanted to say that. So this is just kind of, again, a refresher of the enzymes involved in protein digestion. So basically then with digestion, you end up with either free amino acids or very short peptide chains, generally tripeptides, so just 2 or 3 amino acids hooked together. Or the free amino acids. So all of that's happening inside the gastrointestinal tract, so in the middle, in the lumen. And then we need to get those byproducts of protein digestion into the intestinal cells. And so there's basically 2 membranes that those need to cross, the brush border membrane and the basolateral membrane. So there are separate systems for amino acid transport and peptide transport. So the amino acids are absorbed all along, can be absorbed all along the small intestine into the intestinal cells, but it's primarily in the duodenum and the upper part of the jejunum where that happens. So again, when we think about the importance of digestion and absorption from a nutritional perspective, if we have any kind of damage to that part of the small intestine, we need to be thinking about how that might impact the absorption of amino acids. Often what happens is that if there's damage there, we need more of the amino acids present because it's going to be absorbed at a lower rate, not as efficiently. So just something to keep in mind from a health perspective. With the absorption, there are carriers required. However, some of them are passive and some of them are active. So remember, passive isn't going to require energy. Active does require energy. They do require a carrier, but it may be passive or active. And many of them are sodium dependent. So sodium is very involved in many reactions in the body, and most of these carriers are sodium dependent. Again, the peptide transport has a little bit of a different, a similar type of system, but it's a different system. So there's different carriers involved for the transport of peptides. This graphic gives an image of amino acid transport into the cell. So in the middle here is the inside the cell. So we're looking at an amino acid that's starting outside of the cell. So maybe in the lumen of the gastrointestinal tract. Our body has digested it, broken it down into that individual amino acid, and now it's trying to get it into the cell. And so really what I want you to get from this image is this idea that there is a carrier, a transport mechanism that gets it across the cell membrane. It doesn't normally just float through. Now, that's not to say it can't. Sometimes they do sort of pass through without the carrier, but the normal mechanism is that it would use this carrier. So that's one thing I want you to see from here. The other thing is remembering that many of these transporters are sodium dependent. And remember the sodium dependent issue, or if a carrier or transporter is dependent on something else, often there's this exchange. So something goes in, and you see that over on this side of the image, but something comes in and something else goes out to keep the balance there. So this is a sodium dependent transporter of the amino acids that needs a carrier to get through the cell membrane. It doesn't just float across. So just kind of keeping in mind the different pieces of the picture that are involved in actually getting the amino acid from the gastrointestinal tract into the cell, or from the blood into another type of cell. So as we look at other things, we'll talk about different organs in the body that need those amino acids. For example, when the amino acid goes into the liver cell, we have to have a carrier to get it from the blood into the liver cell. So wherever it is in the body we're talking about, it has to have some way to get across that cell membrane and into the cell to then do the function. Or, for example, in the liver may then put it together into some other product that the body needs. So here is a picture of an example looking at peptide transport. So remember, from digestion, there can either be broken down all the way to amino acids, and we just looked at the amino acid transport, or broken down into smaller peptides. And again, remember, those are generally just di or tripeptides. So now we're talking about very short chains of amino acid. So in this picture, you're looking at an enterocyte. So remember, that's an intestinal cell. So the brush border membrane here is what's going from the lumen of the small intestine, so basically the inside tube of the gut here on the left that you're looking at, into the enterocyte or the intestinal cell, and then it's going to also need to go across this basolateral membrane in order to get into the blood. So over here is kind of our breakdown product of food in the inside of the small intestine. It's going to go into the intestinal cell to be broken down further, and then out into the blood to be transported around to the rest of the body. So we have systems for each of these different transport. So this PEPT1 is the peptide transporter, and this is pumped in with hydrogen is involved in that, and you can see this exchange back and forth between hydrogen and sodium to keep that balance there, and then sodium potassium on the other side. So again, remembering that there's generally, as something goes in, something else goes out in those transport mechanisms. So essentially what's going to happen if protein was broken down into those di or tripeptides, then those are going to go into the enterocyte, where those will then be broken down into the individual amino acids, which those individual amino acids are what are going to be exported then into the blood to carry throughout the body to carry out the functions that they need to have happen. Again, sometimes, normally this is the process. Sometimes, particularly if there's some sort of illness, inflammation, those sorts of things, remember normally these enterocytes, and my artwork is not good here, but if I was drawing kind of another enterocyte right here with the brush border on this side, that those are really tight together, so not a lot of space for things to slip through. But if that space increases, so there's a more permeable membrane, then some of those peptides may actually slipped through. But that's not the normal function. That's, not kind of what the intent, so to speak, is for how those peptides are absorbed. They should normally go into the enterocyte to be further broken down into amino acids before they head out into the blood. So this slide is essentially just showing, or highlighting that amino acids, again, once they're through the enterocyte into the blood, then they're going to be sent out to other types of tissues. So the liver, they would need transporters into the hepatocytes, so the cells of the liver, or they'd need transported into whatever other cells in the body they're going to be functional in, where they're needed. The gamma-glutamyl cycle is one way that amino acids are transported. So there's an image on that, but essentially just getting the sense that there's a lot involved in that transport, that it doesn't just kind of slip into the cell. There's a whole process with many factors that are required to make the cycle happen.